we'll have a presentation about isoproterenol turnover, normal and hypertrophic mark. And we'll most likely end before 9 a.m. Pacific time. But I will use some of the remaining time to do updates. And again, the conferences are recorded and will be accessible online once we process them. If you have any questions during the presentation, please type it in the message box provided in the application. And also, please mute your mic during the presentation to prevent any background noises. Are there any questions? All right. We'll start the following presentation will be presented by Edward Lau. Edward just recently received his PhD in physiology at UCLA. He has worked at Physics Lab for about 4 years as a PhD student. Today he will be presenting about protein turnover in normal and hypertrophic mark. He will describe a recent large scale experiment on protein turnover dynamics at the site. The turnover rates of about 3,000 in normal and mouse strains were analyzed. Without further ado, Edward, go ahead.
Okay, should I get started?
Yeah.
Okay. Let me know if you can't hear me. So my name is Edward. I'm from Physics Lab. I was told to stand in for today's speaker, so I thought what I could talk about is talk about ProTurn from the perspective of the data sets that we use ProTurn to generate. I think my colleagues talked about the software side of ProTurn a couple of weeks ago. But I'm going to just present on a recent data set that we acquired with the help of ProTurn. So maybe we could discuss what are the kind of experiments that that we could do with the software. So I guess I'll start with a little bit of background. So ProTurn was designed to analyze protein turnover data from deuterium oxide or heavy water labeling experiments. So turnover rates are just the rates of natural replacement of proteins inside the cell based on continuous synthesis and degradation. There are a number of reasons why we're motivated to measure protein turnover. Two of them are disrupted protein homeostasis and degradation are actually found in many diseases, and there weren't a lot of tools that experimentally people could look at them easily before. And secondly, what we found out from the experiments is that turnover rates are oftentimes independent to protein expression. So when you measure turnover rates, you might find some kind of disease discriminants that you're not going to find from a protein abundance quantification experiment. So this is a diagram of the protein turnover cycle. You could see basically if you have a protein pool that is represented here. Do you see my mouse cursor if I move it like that?
Yes, we can see it.
Okay. So you see a protein pool here. It could be reinforced by the synthesis of new proteins, and then protein molecules could also be degraded back to amino acids. And you could see how big your protein pool size is. It's really independent from how fast this cycle is happening. So experimentally, how we would try to find out the rate of replacement is to use any kind of isotope labels, and in our lab we use heavy water. So on the top here you could see a cartoon of how the dots they represent heavy labeled amino acid and proteins, and they'll get incorporated. The amino acids get incorporated in the protein over time until the amount of isotopes inside a protein pool reaches the plateau. And if we could measure the amount of isotopes at each time point, then we could plot our kinetic curve shown in the rectangular box in the middle here. And from this curve, we could fit to a kinetic model and find out what the half-life of proteins are. So one thing about ProTurn is that it's specifically designed to handle heavy water labeling. So in figure B here, you could see the difference between let's say N15 or heavy amino acid labeling on the top here, where you would essentially create a new cluster of peptides that you could see in a mass spectrometer, and then you could quantify the amount of new proteins rather easily by just comparing the intensity of this new cluster to the old cluster. In heavy water labeling, because we introduce a rather small amount of heavy water to the peptides, and the mass shift is not that drastic, and what happens is the cluster is going to shift in shape gradually, and ProTurn is what ProTurn does is to integrate each of these individual peaks and then to figure out how much exactly new isotopes and new peptides are in the sample. So some of the questions we want to ask, with these data are, what are the turnover rates of protein in normal and diseased hearts? And we also study a number of different mouse strains in the laboratories. These mouse strains have different susceptibility to our disease models of hypertrophy, and we want to know whether they have different protein turnover rates. And lastly, we want to know what protein turnover could tell us about proteome remodeling, how the proteome changes in the course of disease development. So this is a cartoon of an experimental schema of a heavy water labeling experiment. So we start with 6 mouse strains on the left here, and we will feed them with heavy water for up to 14 days. And at the same time, we also give some of the mice isoproterenol,
yes,
which causes hypertrophy.
Thank you very much. Great. Excellent.
And we would sample proteins in the heart at different time points for kinetic modeling. So how we handle the samples shown here, we would extract proteins from the heart and digest them with trypsin, and then we would separate peptides with 2-dimensional LC and measure an orbitrap mass spectrometer. And after that, we will analyze both the turnover and the expression, where the turnover would be represented by the linear shift of the isotope envelope from the left to the right, whereas the expression is represented by the absolute intensity of the isotope envelope. And we're going to fit each data point to a kinetic model using ProTurn. So I don't think I will go into detail with how ProTurn works, because I believe my colleague presented on it a couple of weeks ago. But basically, it reads in your protein ID list from a search engine result like Prelucid, and then integrate the isotopomers and calculate the fractional abundance of each isotope peak, and then perform nonlinear fitting and output the result in a text format. So I'm going to quickly just show some of the results from the data set from ProTurn's analysis. The first part is, how does protein turnover rate change in normal and hypertrophic hearts in one of the 6 strains in C57. In total, we acquired the turnover rates in abundance of around 2,000 proteins from C57 from around 11,000 peptide time series in the control heart, and around the same number in the hypertrophic heart after the drug stimulation. One of the things we saw was there was really a rather wide distribution of turnover rates, as you could see in this histogram on the upper left here. That corresponds to at least 2 orders of magnitude. This is a log2 scale on the bottom here. So this is in normal, the gray histogram in normal hearts. And in hypertrophic hearts, as you could see in the red histogram here, there's a linear shift toward faster turnover rates. This is sort of a feature of protein turnover that you could see experimentally that you probably would not see in if you measure protein expression, because there's a normalization of the amount of proteins that you could load in the sample if you measure protein abundance, so you don't really see any difference, significant difference in the distribution of the overall abundance that you could measure. So what are the proteins with elevated?
Could I interrupt you? I'm sorry, I
rates?
just have a quick question.
Oh yeah, of course, please.
I'm sorry, and I just wanted to know this before we moved on. What subproteome were you looking at? I'm sorry.
Here we are actually looking at the total proteins.
Total proteins. So did you spin out the this is all cardiac, so did you do like a low speed spin and then you took the entire supernatant and you didn't fractionate any further, is what you're saying, right?
We actually did do the fractionation. We have 3 fractions. One of them is all the soluble cytosolic proteins.
Okay.
And that fraction is the mitochondrial and probably ER proteins.
Okay.
And lastly, we have debris fractions that contains a little of everything, but is enriched for nuclear and extracellular proteins that are not very soluble.
That's why I'm asking, because the sarcomeric proteins would be in that first low-speed spin, and that's kind of, as you know, what I'm interested in. So, okay, thank you.
Oh, thank you, but sorry I wasn't making much sense, probably.
No, that's okay. That's okay. I was interested in what proteins you're actually looking at here, so thanks.
So in this analysis, we recombine all the fraction after we ran the experiments individually. And well, so the first thing we did was, with the data set was maybe an enrichment analysis, such as using Webb-Gestalt and NCBI-David to try to find out what are the proteins with, extreme changes in both turnover or abundance. I'm going to skip this slide of details, but well, we see a lot of extracellular matrix proteins with higher turnover, which is what you would expect in cardiac remodeling, actually. And but one of the things I want to point out would be that you could see here. This is, I guess, sort of like a histogram, a distribution of the ratio of changes after the drug treatment. On the left is the ratio of turnover rates, and on the right is the ratio of abundance of the same proteins, and you could see different proteins that change in each of the 2 attributes. And the extreme changes are enriched in different categories. Extracellular matrix proteins have elevated turnover, and we see a lot of lipoprotein proteins with lower turnover. And then with abundance, a lot of them are microRNA targets that actually have lower abundance or expression after the drug treatment. So this is just a detail of some of the enrichment analysis, and I'm going to describe it. We also tried to plot out in a scatter plot, the 2 different parameters of abundance and turnover to see whether they're proteins with decreased turnover but increased abundance. And these proteins, they're a small subset, but they're enriched for different categories with mitochondrial complex 2 proteins. And this is the graph from the enrichment analysis in Webb-Gestalt. So one of the things we want to ask was, were these decrease in protein turnover specific to the substrate, or was this simply because of a global decrease in protein degradation in the heart? So we did the comparison by giving mice another drug called epoxamycin that basically stops most of the global protein degradation inside the heart. And with the drug, what we could see was that we could measure almost all of the proteins. At least 83 of them actually have slower turnover rates, so they're being replaced at a slower rate inside the heart after the drug. So a second sort of analysis we did was whether there was a relationship in the turnover rates of functionally related proteins. So it's well known that interacting partners, or protein-protein interaction pairs, tend to have similar abundance inside the cell. And we want to know whether they also had similar turnover rates. So we queried around 7,500 protein-protein interaction pairs from a number of databases through Cytoscape, and we combined the networks. And we wanted to measure the average distance in turnover rates between protein-protein interaction pairs. And then we compare that distance to the difference in turnover rates among 10,000 random protein pairs. And basically what these histograms shows are just the average of the ratios of turnover between two interaction pairs, which tend to be a lot more narrowly distributed than random pairs on the right. And you can see the density on the graph to the right here. And what that tells us is that interacting pairs do have more similar turnover rates than random pairs. And we also did the same analysis in a number of different scenarios, such as after the drug treatment, or trying to control against the interference of abundance by only selecting a subset of proteins with similar abundance and doing the same analysis. And this graph is just another way of looking at the same analysis. The orange lines, and well, first of all, the proteins that we measured are aligned along the circumference based on their chromosomal location. And then the orange lines in the middle are protein-protein interaction pairs that involve at least one protein in chromosome 1 that are also between two proteins with turnover difference of 1-fold or less, whereas the gray lines on the right are between protein-protein interaction partners with more than 1-fold difference in turnover rates. So if protein-protein interactions were orthogonal to turnover, then you would actually expect to see more lines here. But the vast majority of protein-protein interaction pairs were actually between proteins with very similar turnover rates. So lastly, just to, and we wanted to look at, we wanted to compare the turnover in all 6 strains that we studied, and we're still, doing the analysis and see what are the best ways we could look at these data. But one of the things we did was, well, first we wanted to ask, do we even see any turnover difference between different strains? And secondly, do they correlate with the severity of the heart disease after the drug treatment in the strains? So this is just some of the measurement metrics with the mass shift and the goodness of fit and standard error of our experiments. And this graph shows the distribution of turnover rates in all 6 strains, and they tend to be quite similar actually, despite what the graph shows. But the individual proteins would be would have difference and be poorly correlated. And this just shows the different responses of the 6 strains to the same drug. For example, one of the metrics you could use to measure the response to the drug is how big the heart grows in size, and you could see clear differences. Some of the strains on the upper right here are more susceptible to the drug. So initially we were just going to do some rather straightforward enrichment analysis to see if there are proteins with higher turnover in one strain versus another strain of mice, and whether they are functionally related to any categories. Well, one of the categories that was only enriched in one particular strain was these sodium-potassium exchange channels in the sodium-potassium channels in the heart. And you could see clearly that the turnover rates increase after isoproterenol only in one particular strain, but not so much in the others. So we wanted to cluster all the data together and see where we could discern any structure in the data, any patterns that we could see. So we used a package in R called WGCNA, which stands for Weighted Gene Correlation Network Analysis. It's quite similar to clustering, I think, if given the size of data that we have. But basically, what it does is it compresses. We have around 2,000 comparisons among the 6 strains. The package compressed this data into maybe 15 modules or clusters of similar behaviors. So we can now just take these individual clusters to do correlation comparison with the traits, and we won't be, it sort of relieves the burden of multiple testing. Instead of doing 2,000 hypothesis testing, now we're doing 15 of them using, clusters rather than individual proteins. So these clusters in the package, they color-coded them with different names of different colors. And you could see here, this is a heat map of the correlation coefficient when we take each cluster and we try to do try to analyze the correlation of each cluster with the heart rate at different time points after the drug treatment. And we see there are 3 clusters that seem to be clearly correlated with the degree of, with the heart size or degree of hypertrophy after the drug treatment. So we were interested in knowing what they were. So these clusters were named turquoise, yellow, and purple. And so you could see here on the right, the membership of a protein to a particular cluster. It's correlated to how significantly its turnover behavior in all 6 genes is correlated to the heart rates of the 6 genes. And this module also overlaps, on the left here, you can see it overlaps quite significantly with existing protein-protein interaction networks. So they seem to be a module, or a module of proteins with similar turnover that behave similarly after the drug treatment, and also is correlated with the severity of the response inside the mouse. And these proteins are enriched in a number of things, including a lot of sarcolemma proteins, actin filaments, and also some microRNA targets. And I plotted out the correlation of some of the proteins here. It includes a lot of glycolysis proteins. So this is only a simple correlation analysis, and we're trying to look into some regression models with them to take into account some of the covariates now. But at least here you could see that in this particular population, it seems like the higher the turnover rates of some glycolytic proteins, usually the more severe the heart disease is after the drug treatment. And this is different from the behavior in another module, yellow module, which is enriched for other proteins, including beta oxidation proteins and mitochondrial inner membrane proteins. And these proteins have sort of the opposite behavior. They tend to have lower turnover in the mice with more severe heart diseases. So what we saw was there were 2, I guess, the 2 metabolic cluster of proteins that behave differently in terms of the protein turnover. And we couldn't see that from the protein abundance data, as I think I'll show in a slide later, because when we also measured protein abundance, there's no correlation with the severity of heart disease. So this is an observation that I guess you could say it was unique to protein turnover rate after analysis from ProTurn. There are other modules that have interesting behaviors, but they don't really map to any known protein-protein interactions or known functional categories, and we're trying to figure out whether these proteins are actually related in terms of function. We also have measurements, at least from our collaborators, of other cardiac traits, rather than just in addition to just how big the heart is growing after a drug treatment. These are measurements of cardiac functions, and we saw other modules of protein turnover data that correlate with these cardiac traits. And as I said, lastly, we tried to also build a similar network clustering with the abundance data, but we saw something very different, and there was no clear correlation between the abundance of these proteins with the turnover rates. So I'm going to wrap up here, and I guess the take-home message is I just want to show these are some of the experiments that we use ProTurn for, and it'd be good if we could we could discuss on whether there's any ideas about what are the kind of analysis that we could do, and what are the other things that we might want to add to ProTurn to support similar kinds of experiments. So I'll be happy to take any questions, and thank you for listening to me.
Thanks, Edward. That was a great presentation, and we're really grateful you could present on such short notice.
Hey, Edward, this is Sarah.
Yeah, hi, Sarah.
Hey, great presentation. It's really nice analysis. It's very helpful. And just a comment on when you were describing your sort of protein behaviors, the turquoise, the yellow, and the whatever the other color was.
Right.
I think you said that you detected that some of the fatty acid oxidative enzymes showed decreased turnover rates in isoproterenol, chronic isoproterenol treated hearts. Is that correct?
Right. That's correct.
That's interesting, and I think it'd be really interesting to relate some of those metabolic changes to what's actually happening with substrate utilization in the mitochondria in heart failure, because the mitochondria go from a primarily fatty acid oxidative state, in a normal heart, to in heart failure it switches to more of a glycolytic state, which is, that's been proven over and over again. So it would be interesting to see how the sort of oxidative, fatty acid oxidative enzymes, their turnover rates compare with those involved in like glycolysis. Because then you could potentially link that to the changes in substrate utilization. I think that's very interesting. What was the other?
Right. Yeah. That's also what we're thinking. You're right. I didn't get to go into that, but definitely, as you said, the substrate utilization between the two, metabolic clusters, like glycolysis versus fatty acid oxidation, that is very well established. And it seemed to me, at least, what the data said was you could see a sort of molecular basis of this kind of behavior in terms of turnover that you don't really see in abundance. But of course, it might not be, actually the causal factor of why, substrate utilization could be different, because there's always the availability and import of the substrate that could change. But definitely, there is a correlational relationship, and we thought it was really interesting that, you could see that from isotope labeling experiments.
Yeah, I like that a lot. And I wonder, well, if there are changes in abundance, I wonder which happens first,
Right.
Kind of, because we've always sort of thought that protein dynamic changes may herald changes in protein abundance. So it would be sort of neat to look at that too. But I had one other comment or question. Back to the protein interaction data, did you look like, I know you were sort of giving us a global assessment of all the proteins you looked at, and those graphs were very cool, but in complexes that you know a lot about, like just take one of the respiratory complexes, did you look at all of the individual subunits that you know constitute one of those macro complexes? And did you detect any patterns regarding, were there a couple subunits that perhaps turned over rapidly and could, or, were distinct in their turnover rates relative to the rest of the proteins in the complex, and therefore you could possibly think about whether or not those subunits were rate limiting for complex formation, or, do you see what I'm getting at?
Yeah, we looked at that before. I guess if I were to summarize, what we saw was some complexes are a lot more tightly kind of correlated in terms of the abundance and turnover of the subunits, like the proteasome. Each of the subunit tends to be very tightly clustered in terms of the turnover rates. Some complexes, especially the larger complexes.
I'm sorry, with the proteasome, is that just 20S subunits, or is that the associating regulatory particles as well?
Oh, right. It's actually just the 20S.
20S, yeah, that's what I would expect.
The 19S tend to have faster turnover rates, actually. Some of the larger protein complexes, like the ribosome and MAB mitochondrial complex 1, the subunits tend to have a wider distribution of turnover rates. And we definitely we looked at it before for complex 1, and some of the assembly factors tended to have faster turnover rates than the core subunits, especially the ones that are encoded by the mitochondrial genome. And we were wondering whether you could use turnover to look at the assembly, and it seemed to me like maybe the subunits are less tightly associated. Like the assembly factors, you might not be constantly associated with the material complex. They tend to turn over faster. But it's a I think there's some noise in the data if you just look at it very individually, and it's hard to be very specific about the conclusion unless we have very large data sets from a lot of samples. But I think, to me personally, what I saw, there's definitely hints of that, there.
That's exciting.
Thanks for the comments.
Yeah, thank you for your talk. It was great.
Oh, I guess I have a question.
Yeah.
So I mean, I know we do a lot of analysis in R, and I know a lot of researchers use things like MATLAB and Python and so on. Is there any way, from the point of view of somebody who uses ProTurn for research, is there any way that you could see yourself benefiting from more interconnectivity with other software? I mean, other than just getting like file formats to be consistent for different the output of one tool versus the input of another, is there any way you see
Right.
ProTurn or similar tools operating more closely with existing software?
I think well, in terms of graphics, I think I tended to do the post analysis, I guess, if you will, in R simply because of the graphical capacity. It just gives nicer looking graphics, I guess, than what we had in Java. I don't know if it's something that we have interest in, or whether generally overall in all the tools, whether you want to export scripts to visualize the data in R and do everything in Java. But yeah, I think that the reason is mostly graphically. I have a lot more flexibility.
Right.
It's a great presentation. So as of now, I'll just give some quick updates before we end our conference. So this weekend is daylight savings in the U.S. So our time will spring forward an hour. However, in the UK, the times will they don't move up an hour until March 29th. So for the next two conferences, March 13th and March 20th, those conferences will be an hour later in the U.S., but of course remain the same in the UK. And March 27th is a holiday in California, so we will not have a conference that Friday. So therefore, in the next 2 weeks, the conferences will start at 8:30 a.m. Pacific time, and the presentations will begin at 9 a.m. and end at 10 a.m. Pacific time, and we'll have an open discussion period from 10 a.m. to 11 a.m. Afterwards. But I'll be sure to mention this in Monday's email, just in case. But are there any questions regarding the time change for the next 2 weeks? Okay. And also, I just want to go over the GitHub page.
Yeah.
Okay, so if you look at the screen, so I have the GitHub page for our WGCNA Center, and it contains a list of repositories. Currently there's only a couple, but so if you click on APIs, you'll see that the expression interface is on there, and then if you click on it, it'll show you the expression interface page. And then if you go to UCLA, click on CovaKD's repository, it'll link you directly to the actual code repositories. But in order to access some of these repositories, some of them are private. So, for example, if you're not already logged in, click on the repository for CovaKD, it will give you this 404 error because you're not part of, or you weren't invited, and you don't have access to the repository. So what you need to do is, in order to gain access, fill out this form, right? And then once you fill it out, hopefully within a couple days, I'll give you access to all the GitHub repositories, so you can browse around, have access to them. And if also you would like write access and you'd like to be included on a project, then please feel free to email me. And it also helps if, so I'm in the process of gathering all the repositories to post on this webpage. If you can go up here and fill out this repository submission form, that'll help a lot so that I can easily add your repository to the website. Yeah, are there any questions about the GitHub webpage? And also, so we'll be also using Trello in tandem with GitHub. I know a lot of projects are still in the early stages of it, so won't have to do as much, but hopefully we'll start using it more. But basically, eventually each page should have some information about the tool. And also contact information. And I guess current progress. So the status of the project is. And then the other stuff can be all, all the collaborations that are happening. For example, if we're working on the expression interface API, then we would, have like a card, and then we'd have like a to-do list and assign, each member a to-do, so that everyone can see what everyone else is doing, and then when you complete it, you just cross it off, and everyone see that it's been done. But yeah, I'll be contacting. I've already contacted several groups regarding the Trello usage, and I'm hoping to work more with you guys in the following week to really start getting Trello implemented and start being used. Are there any questions?
So I have a question. Do I need an invitation to join Trello, particular Trello board, or is there like one board for each tool? And if there's a
yeah, you would need an invitation. So feel free to email me, and I'll invite you to this organization right here. So this is the B2 project organization board, and as you can see here, there's a list of all the tools involved in the B2 project. Yeah, so email me first. I'll invite you to this organization, and you'll have access to all these boards, the information.
Oh, okay. Thanks.
Any other questions? Okay. If not, we'll be here until 10 a.m. Pacific time for any further questions. Yeah, thank you all for attending. I'll be giving up an update on Monday for next week's presentations, and also some of the updates and reminders for the conference. Thank you all.
Thank you.
Thank you.